Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about to investigate the analgesic properties of morphine in mice using the tail flick method. At the end of this video, we will be able to tell about the analgesic effects and properties of morphine by using it on mice through the tail flick method through the analgesiometer instrument. Now let us start with the video. At first, let us see the introduction. The introduction part says pain management is crucial and morphine, a potent analgesic, plays a vital role. Our study employs the tail flick method in mice to assess morphine's pain relieving properties. So the statement says that pain management is very crucial and morphine is an analgesic, potent analgesic drug and it plays a vital role in the pain management. And our studies is based on this pain management and the effects of morphine on the pain management. To assess by the tail flick method, by using a rat, we are going to use the tail flick method of through mice to assess the morphine's pain relieving properties. Now let us see the equipments required for this experiment. At first, we require the apparatus analgesiometer and mice cages. Then we require our subject on animal is mice, which should be of between 25 to 30 grams. This is the average weight of the mice. Then drugs. We require the drug morphine sulfate, which should be of dose 5 milligram per kilogram. We have to prepare the stock solution containing 0.5 milligram per milliliter and inject 1 milliliter by 100 grams of the body weight of the mouse. We have to administer the dose according to this. Now let us learn more about the analgesiometer. Analgesiometer, as we can see, this is a diagrammatic representation and the image of the analgesiometer. As we can see here, these are the switch on and off buttons. This is the meter and here we have the cage. As we can see here, this is the cage where we will keep our mice. We will insert the lid. We will open the lid from here. It opens and we insert our mice and the mice, the face of the mice should be here and the tail should be here. And this is the heating mantle where the tail of the mice is kept and when it heats, the tail is flicked by the mouse. And that's how we perform the tail flick method. The introduction of this instrument says this instrument is used to evaluate analgesic effects of drugs in experimental animals against the thermal stimulus. So, this instrument is used for the to evaluate and study analgesic effects of drugs in animals. And by the thermal stimulus. Thermal means heat, and stimulus is the reaction of according to the condition. So thermal heat is the condition, and stimulus is the response for that. And through that, tail flick method is performed. The principle of this instrument is that the principle says that it works on the principle of observing the pain threshold in rodents before and after the drug administration. So, principle says that we have to observe the pain threshold in rodents, means the pain that can be bared by the animal that is observed before and after the drug administration, which means before administering the drug in a normal condition, how much pain can the animal bear, and in how much pain it flicks its tail, and even and after the drug administration, we observe the same thing in by feeling how much pain the animal flicks its tail. So we observe and perform the drugs, the pain management drugs, or we can say analgesic drugs. And we study about that, how much the drug shows its effects. Now let us move forward in our experiment. Let us see the principle of this experiment. The principal part says that pain management is a critical aspect of both clinical medicine and scientific research. The chronic pain affects millions of individuals worldwide, necessitating the development of effective analgesic agents. Morphine, a powerful opioid, has long been recognized for its potent pain relieving properties. However, understanding its precise mechanisms of action and assessing its efficacy in animal models is essential. Our study aims to investigate the analgesic effects of morphine in mice using the tail flick method. So the above para says that Pain management is very critical, is a critical part of the clinical medicine and the scientific research. Chronic pain affects the many individuals in a million of quantity, and morphine is a very powerful opioid drug. Is a powerful opioid drug and has been longly recognized for its pain relieving properties or potent pain relieving. Potent is that it requires a less amount. It is a very strong drug. However, understanding its precise mechanism action and assessing its efficacy in animal models is very essential. So we have to understand it. How does the morphine works on animals? So for that, we have to perform this experiment. Our study aims to investigate the analgesic effect of the morphine in mice using the tail flick method. We have seen that what is tail flick method. Now we induce pain in experimental animals using different methods. We can induce this pain by different methods. The methods are that first is the thermal pain. We apply radiant heat to their tails, causing them to withdraw. Tail flick method. This heat mimics a painful stimulus. So we are performing this thermal pain method, where we apply heat to the tails of the animal, which causes them to withdraw. Withdraw means they move their tail from that to avoid the pain. And the heat mimics a painful stimulus. This is the tail flick method. Second one is the chemical irritation. Chemical irritation says that we use irritants like acetic acid and bradykinin. These substances provoke discomfort. So for that, we use acetic acid, as we know that is a very highly reactive acid, and it is a very strong acid, and bradykinin. So we put these acids and chemicals on the tails or on the animal, and this substance provokes discomfort to the animal, and that's how we induce the pain. Then physical pressure. Physical pressure, we compress their tails, or this pressure elicits a painful response. In physical pressure, we compress their tails, which means we have to press their tails and compress their tails so that pain is induced in their tail, and this how a painful response is observed in the animal. Now let us see some key terms and concepts. First is analgesia. What is analgesia? The definition says analgesia refers to a relief of pain or a reduction of pain sensitivity. It involves blocking pain signals or altering pain perception. Significance. Morphine is a classic analgesic used to manage moderate to severe pain. So analgesia is the relief of pain or reduction of the pain sensitivity whenever we are feeling pain, and if we administer any pain relieving drug. So that's analgesia when the relief of the pain is felt or the reduction of the pain sensitivity is felt. It involves blocking pain signals or altering pain perceptions. How does it work? It blocks the pain signals or alters the pain perception. And for that, morphine we use that it is an analgesic drug. It is a classic analgesic used to manage moderate to severe pains also. Then morphine. Morphine is an opioid alkaloid derived from the opium poppy, Papaver somniferum. This is the scientific name of the opium poppy. It acts as a primarily on mu opioid receptors in the central nervous system. So morphine, as we know, that is an opioid alkaloid, and it is derived from the plant opium poppy, whose scientific name is Papaver somniferum. Then it acts on the mu opioid receptors. It is the muscarinic opioid receptors that is present in the central nervous system inside the body. How does it work? Mechanism of action. Morphine binds to muscarinic receptors, inhibiting neurotransmitter releasing and reducing pain perception. So morphine binds to these receptors, which inhibits the neurotransmitters and inhibits the release of the neurotransmitter, and then it reduces the pain perception. Clinical use of morphine. It is widely used for acute and chronic pain management, as we know, especially in cancer patients. Okay. Now, tail flick method. Principle of this method says that the tail flick method assesses nociception pain perception in rodents. A focused heat source is applied to tail, and the time taken for the animal to withdraw its tail flick latency is measured. So, this method is used for the pain perception in rodents to study about the pain perception. We call it nociception. And in this method, we focus the heat source applied to the tail, and the time taken to withdraw the tail of the animal is measured. The withdrawal of the tail is also known as tail flick latency. Rational. Increased latency after drug administration indicates analgesic activity. So, whenever a drug is induced, an analgesic drug is induced, then the increased latency after that indicates the analgesic activity. So the drug will increase the latency or time taken to withdraw the its tail is increased. Next is the animal model. Mice. Choice of species. Mice. Mus musculinus. This is a scientific name of mice, and this is commonly used due to their genetic similarity to humans and ease of handling. As we know, mice are small animals. They weight very less in they very less in weight. According, we can say that we know that its average weight of a mouse is 25 to 30 grams, and the genetic characteristics are similar to humans, and that's why they're easy to handle. Weight range. Select mice weight between 20 to 25 grams to ensure consistency. Experimental setup. Now, the setup that we are using to perform the experiment. First is radiant heat source. We require a radiant heat source. A controlled light beam delivers heat to a specific spot on the mouse's tail. Then, baseline measurement. Determine the tail flick latency before drug administration. We have to determine the tail flick latency. So we have to perform before administering the drug and after administering the drug. We have to perform it 2 times so that a baseline is measured. Drug administration. Administer morphine intraperitoneally at a dose of 5 mg milligram per kilogram. So we have to administer that our analgesic drug morphine through the intraperitoneal route. Intraperitoneal route is that route where we inject our drug through the syringe into the abdominal region of the animal. We will study it later while performing the experiment. And the dose should be given of 5 milligram per kilogram. Post administration measurement. Post administration measurement. Record tail flick latency as specified intervals 15, 30, 45, and 60 minutes. So we have to perform this experiment on the tail flick latency at 15 minutes at the latency and intervals of 15 minutes, 30, and 45 minutes. Now, research objectives. Our study seeks to evaluate the analgesic effect of morphine in mice, establish a dose response relationship, and understand the temporal profile of analgesia post morphine administration. So, these are the objectives that we have to accomplish in this experiment. This is the ideal observation table of this experiment. As we can see, this is the serial number. So, we are using 6 mice for this experiment. Here, this column shows the body weight of the animal. This is the basal reaction time, which means the tail flick latency before administering the drug. We have to perform it. We will take 3 rounds for the mean to calculate the mean. Then this is the mean basal reaction time after morphine administration. So this is the mean time after the drugs administration at 15 minutes interval, at 30 minutes, and 60 minutes interval. And for this also, we will perform 3 rounds. Before administering the drug, we will perform 3 rounds, and after administering the drug for each interval, we will perform 3 rounds. For 15 minutes, we will perform 3 rounds. For 30 minutes, also 3 rounds, and for 60 minutes, also we will perform 3 rounds. This is the main chart or the basal reaction chart, we can say. Now, let us see the procedure of this experiment. At first, we have our animal preparation. We have to prepare our animal. We have to weigh and number the mice. Then we have to record the weight of each mice and assign a unique identification number to each mouse for tracking. Now then, baseline reaction time measurement. We have to perform tail flick method. Place the mice, place then the last and place the last 1 to 2 centimeter of the mouse's tail on the radiant heat source. So when we place our mouse on the analgesiometer inside its cage, then the tail of the mouse should be placed on the radiant heat source for at least 1 to 2 centimeter long. Now, tail withdrawal response. Observe the mouse's behavior. Normally, a mouse withdraws its tail within 3 to 5 seconds due to the heat. Set a cutoff timer, for example 10 to 12 seconds to prevent tail damage. Any mouse failing to withdraw its tail within 3 to 5 seconds is excluded from the study. Now, after that, confirm normal behavior. We will take 3 to 5 basal reaction time measurements for each mouse. Space these measurements at 5 minutes interval. Ensure that the mice exhibit consistent and normal responses. We have to perform each round and we have to give them 5 minutes interval between the each rounds. Now the third is the drug administration. We have to administer our drug morphine through the injection. We will administer morphine sulfate intraperitoneally. through the interperitoneal route to the mice. Then for that we will use a dose of 5 milligram per kilogram, adjusted based on the experimental requirements. Okay, and then note the exact time of the drug administration. Post administration reaction time management. So after administering the drug, we will observe the mice at the following time intervals after morphine injection. We will observe the intervals of 15 minutes, 30 minutes, and 60 minutes after the drug administration. And for each interval, we will perform 3 rounds. 3 rounds for 15 minutes, 3 rounds for 30 minutes, and 3 rounds for 60 minutes. Maximum analgesia. As the reaction time reaches 10 seconds maximum analgesia, remove the tail from the heat source. This prevents tissue damage while ensuring accurate measurement. So, after the drug administration, if the time reaches more than 10 seconds for the tail to flick, we have to remove the tail from the heat source to avoid any damage to the tissue while ensuring the accurate measurement. Now, let us perform this experiment. And for that, we will perform our experiment in our RJPT simulatory laboratory. As we can see here, this is our lab setup. On the left side of this lab, we can see the cage and the weighing machine. This left side toolbar contains the cage and weighing machine. When we will click it, the cage and the weighing machine will be placed on the table. On the right side of the toolbar, we can see the syringe, which is pre-filled with the morphine drug. At the bottom side, we can see the instruction part, which says and shows the procedure part, and through that we will perform our experiment. On the right side of that, we will see our next and skip button, through which we can directly move on to the next step and procedure. On the top side, we will see our observation drop-down table. When we will click it, we will see this is the ideal observation table of our experiment. This shows the serial number 12356. We are using 6 mouses, then the body weight of each 6 mouses, then the mean weight, then the basal reaction time of the first round, second round, and third round. We will perform the tail flick method for 3 rounds for each mice, and then we will calculate the mean of it. Then after the drug administration, we will see the basal reaction time for 15-minute interval, for 30 minutes, and 60 minutes. And for each, we will perform 3 rounds. While clicking this procedural flow button, we will see that this is the procedural flow chart. These are all the steps that we are going to perform while performing this experiment. These are all the instructions that are going to be shown in this instruction side. We can directly click to any button and jump to that step directly. Now let us perform this experiment. The first instruction says that place the cage from the left sidebar. We will click this cage button, and as we can see here, this is the control group's cage, and inside of that, 6 mouses are placed. After that, the instruction says place the weighing machine from the left sidebar. We will click this weighing machine, and as you can see, the weighing machine is placed over the table. Now weigh each mice. Now we will weigh each mice. So we will click the start weighing button. As we can see here, we are withdrawing a mouse, and we are marking it with the single line on its tail, which indicates the mouse number 1. On this, we will draw 2 lines. As we can see, 2 lines are drawn, which shows the second number of the mouse. Now, for third mouse, we will draw 3 lines. We will see here 3 lines. And now we will weigh it. It is of 23 grams, which means it is an average mouse. By clicking this observation table, we can see the body weights are being written directly into the observation table. After weighing it, each body weight is being written over here, as we can see here. For this is the 5th mouse. We will draw 5 lines, 3, 4, and 5. As you can see, 5 lines, and we will weigh it, and it's of 24 grams. And this is our last mouse. We will weigh it. We will draw 6 lines to denote the 6th number of mouse. It is of 25 grams. Now we will place it again inside it. These are all the 6 mouses body weight. Now we will put the mice in the analgesiometer for reading. So now we will perform our tail flick method. As we can see here on the top side, shows that the round 1 for checking the basal reaction time of the control group. This is the round 1 for the control group. On the left side of the instruction, we can see the stopwatch. The stopwatch We'll count, we'll start, and we will observe the time for the tail flicking of the mouse. Now, the instruction says put the mice in the analgesia for reading. So we will take the mice and place it inside the analgesia. As we can see here, this is our analgesia as we have seen as we have seen earlier. This is the case, and this is the thermal heat source. This is the on and off switch. So now we will switch on the machine for reading. We will click the switch on button. We will switch it on. As we can see, the tail is flicked. The tail is withdrawn of the mouse, and the stopwatch has recorded 3 seconds. On the observation table, we will see it has shown 3.5 seconds. Now we will take another mice. We will place the second mice inside the analgesiometer. Now we will switch it on. As we can see, the stopwatch has started and the tail is withdrawn due to the heat stimulus. Now we will take our third mouse by clicking the take mice button. We will take our third mouse and place it inside the cage of the analgesiometer, and then we will close the lid so that the mouse does not move. We will place the tail on the heat stimulus, and now we will switch it on. As we can see here, the tail is withdrawn due to the heat stimulus. Now we will go on for the fourth mice. We will place it inside the cage and then close the lid. Now we will switch it on. And as we can see here, it has shown the withdrawn. Similarly, we will perform it for the fifth mouse. We have to perform this experiment very carefully so that the mouse does not get harmed. We have to take care of the mouse very carefully. And we have to operate this analgesiometer also very carefully. We have to observe that the tail does not get stick to the heat source. Otherwise, it will damage the mouse's tail's tissue. This is our sixth and the last mouse. And we are performing this for the round 1. We switched it on and we will observe the tail withdrawn. And now we will wait for 5 minutes so that all the mouse will get rest. And we can skip this part by clicking the skip button. And now we will move on to the round 2 for checking the basal reaction time of the control group. We will perform it similarly like the first round. We will place for our first mice inside the cage of the analgesiometer, and then we will switch it on, and we will observe our tail withdrawal. Again, we will place it inside the control group, and now we will similarly perform for the next second mouse. By clicking the next button, we will directly move on to the next mouse or to the next step. As we can see here, by clicking again the next button, we will move on to the next step, which is the switching on of the analgesia meter for the second mouse. The tail withdrawn is observed, and by clicking the skip button, we will directly move on to the next whole step. So we will perform it for the third mouse. So by clicking the skip button, we can see that it has moved to the next step directly for the third mice, and now we'll perform it similarly for the fourth mice. By clicking the skip button, we can directly jump to the next step very directly. And now we will again wait for the 5 minutes to perform the third round. We can skip this part, and now we have reached to the third round. We will place it gently inside the cage of the analgesiometer, and we will switch it on. As we can see on the observation table, this is the second round's reading for the basal reaction time before administering the drug. And this is the third round's reaction. We can skip this part also by clicking the skip button. So we'll perform it similarly like the first and the second round. And now we'll perform it for the our last mouse, for the sixth mouse, and the basal reaction time is 3.5 seconds for the last mouse. Now again we will wait for 5 minutes. And now the instruction says that place the syringes from the right sidebar. We will click it, and as we can see here, these are the 6 syringes for the 6 different mouses. And now inject the drug in each mouse. So now we will apply our morphine analgesic drug to each mouse through the intraperitoneal route. As we can see, one by one we will inject through different syringes to the mouse. We are using intraperitoneal route, which is the abdominal region of the animal. As we can see, this is the abdominal region, and we are directly and injecting our drug to the mouse. And we are using different syringes to prevent any infection reaching another mouse from another. So we'll perform it similarly for all the 6 mouses. We will inject our morphine analgesic drug. And after that, we'll place it again inside the drug group's cage. And after that, we will wait for 15 minutes so that the drug may show its effect for 15 minutes. And for the 15 minutes interval, we will perform our experiment. We will skip this part. And now we will perform the round 1 for checking the basal reaction time of the drug group after 15 minutes. We will perform the tail flick method again. We will gently place the mouse inside the analgesia cage, and we will place its tail on the heat source. Now we will observe the tail withdrawal. One by one, we will perform it for all the mices. Now we can see here, we are switching it on, and this is the tail inside the source, and that we can observe the tail withdrawal. Now for the third mice, similarly, we will place it gently so that the mouse doesn't get hurt, and we have to observe that the tail does not get stick to the heat source, otherwise it can damage tissue of the mouse. Similarly, for the fourth mouse, we will take the mice and place it inside the analgesia cage, and then we will close the lid so that the mouse doesn't run away from the cage, because the heat stimulus can also show in the way of that. Due to the heat, the mouse can run away also. That's why we are closing the lid. We will perform it for all the 6 mouses. Now for the fifth mouse, we will see the tail withdrawal. As you can see, the stopwatch has started. And now we will move on to our last mouse. Now we'll switch it on. As you can see here, it's tail, and it has shown its withdrawal. Now, after this, after round 1, we will wait for 2 minutes. Then we will skip this part, and we will move on to the round 2 of the basal for checking the basal reaction time of the drug groups after 15 minutes. We will switch it on, and we will see the reaction of the tail withdrawal. And similarly, we will move on for the next mice. In the observation table also, you can see this is the mean value of the basal reaction time before drug administration. As we can see here, this is the observation table. This is the mean value of all this basal reaction time. And now we will move on to our next mice. Now we'll move on to the next mice by clicking this next button. We can directly move to the next mouse. We can directly jump to the next step. We will see the basal reaction time reading after the drug administration of the morphine for the third mice. We can see here the tail withdrawal of the trail. And by clicking the skip button, we can directly move to the next step. We will move on to the fourth mice and for the fifth. And now we will record it for the sixth mice for the second round of the 15 minutes interval. Now we have recorded for the second round also for the 15 minutes interval. And now again we will wait for 2 minutes. And now we will take the readings for the third round also for all the six mices. We will skip. We can directly move to the next portion. We will record it for the fourth, fifth, and our last sixth mice. You can see the tail withdrawal technique, tail flick method. And now again we will place it inside the drug group's cage. And after for the 15 minutes interval, now we will wait for another 15 minutes. Why? To check for the round one for the checking the basal reaction time of the drug groups after 30 minutes. We will skip this part. And now 15 minutes are over. And after 15 minutes, 15 plus 15 equals to 30. So now we will check for after 30 minutes. These are the readings, mean readings of the three rounds of the 15 minutes. And this is the total mean of the all the six mouses. Now we will put our mice in the analgesia for the reading of the round one for the 30 minutes interval drug groups, basal reaction time. As we can see here, the time has shown increasing. The timer has shown increasing, which means our morphine drug has shown its effect. It slowly has started showing its effect, and it has shown by increasing the timer in the stopwatch. Before administering the drug, we have observed that the mouses took only 2.5 seconds to 3 seconds, but now it has seen that after the drug administration and slowly after the time passes, there was 15 minutes interval, and after the 30 minutes interval, the timer has increased, which means the morphine drug has shown its analgesic activity, and the timer in the stopwatch has increased to 4 seconds to 5 seconds. And by this also we can understand that the analgesic that morphine is an analgesic medication and drug which has shown its therapeutic activity by analgesic activity, and it is a very potent it has shown it is a very potent drug. Similarly, we will perform it for all the six mouses. Now we will take our another mice. Again, we'll place it inside the cage of the analgesiometer and we'll switch on the analgesiometer, and we will see and observe the tail flick of the animal. As you can see in the stopwatch, and now we performed it for the fifth mice, and now we will do it for our last sixth mice, the round one for the 30 minutes interval. We will click the switch on button. And now we will note the readings. And now we will wait for 2 minutes. And now we'll perform the round two for checking the basal reaction time of the drug group after 30 minutes. We will take our first mice, place it inside the cage, and we will switch it on the analgesiometer. And we will observe the tail flick and tail withdrawn of the mouse. By clicking this next button, we can directly jump on to the next step. As we can see here, we are observing for the second mice of the round 2 of the 30 minutes interval. By clicking the skip button, we can directly jump on to the next mouse. For the third mouse, we have observed, now for fourth, now for fifth, and now for our last mice, sixth. As we can see on the observation table also, this is the reading of the 15 minutes interval and its mean value. The mean value, these are the mean value of the 3 rounds, and then the mean value of this, all the 6 mouses. Now we will have to wait for 2 minutes to perform the round 3 for checking the basal reaction time of the drug group after the 30 minutes. This is the third round. We will perform it similarly like all the other rounds. As we can see here, the timer has started to show that increased time, which means the morphine drug has started showing its activity. By clicking the skip button, we can directly jump on to the next mice, the second mice, the third mice, the fourth, the fifth, and for the last sixth mice. The sixth mice is inside the cage of the analgesia meter, and the heat stimulus, the heat source will produce heat as the tail withdrawal is observed. And now we have completed our third round for the 30 minutes. Now again we will wait for 30 minutes to perform for the 60 minutes interval. We will click the skip button and we will take the mouse and place it inside the analgesia meter for the round 1 for checking the basal reaction time of the drug group after 60 minutes. As we can see, the timer has increased. Similarly, we will record it for the second mice. We will switch it on the analgesia meter. And now the third mice. We will switch on and we will record it. All the readings are being recorded in the observation table. By clicking the next button, we can directly jump on to the next mice. This is our fourth mice. And similarly for the sixth mice. We have to carefully perform this experiment so that the mouse doesn't get harmed. The heat source can get stick to the mouse's tail and it can damage its tail. So we have to carefully place the mouse inside the analgesiometer and stick its tail to the heat source. As we can see here, heat stimulus has shown and it has withdrawn its tail. The timer has been increased and now we will wait for 2 minutes to perform the second round for the checking the basal reaction time of the drug groups after 60 for the 60 minutes interval period. We will place it inside the mouse cage and then we will switch it on. You have to place the tail on the heat source. We will observe the tail flick. By clicking the skip button, we can directly jump on to the next button. As we can see here, this is the 30 minutes interval times and the 3 rounds mean values. These are all the these are all the values of the mean value of the 3 rounds of the 3 rounds that we have performed for each mice. And this is the total mean value of all the 6 mices. These are the values. The first mice is shown 8 seconds, then 6.5, 7, 8, 7.5, 8, and the total mean value of all the 6 mice is 7.4. It has shown that it has increased value. By comparing it with the by comparing it with the 15 minutes interval, we can observe that the 30 minutes interval timer has been increased, which shows that the morphine has shown its therapeutic activity of the analgesic activity. By clicking the skip button, we can directly jump on to the next step or directly jump on for the next mice, for the fifth and the sixth mice. And now we will wait for 2 minutes to perform for the third round for checking the basal reaction time of the drug group after 60 minutes. Now we will switch it on and we can see that the stopwatch has started and the tail withdrawal has been observed for the first mice. By clicking the skip, we can also jump on to the next step as we have performed similarly for all the mouses, for all the rounds and all the time intervals. And now we will check for the sixth mice reading for the third round of the 60 minutes time interval. As we can see, we have observed our tail flick and again we will place it inside the drug group. Now again we will wait for 2 minutes and now we have performed our experiment. We have completed our experiment. This is the mean value for all the three rounds of the single mouse and these are all the mean values of the all the three rounds for each mice. And this is the total mean value of the all the six mouses for 60 minutes interval. As we can see, it has shown an increasing order for the time interval or the time for the tail flicking. Now we will see the result part. The result shows that the tail flick latency, which means the mice administered morphine exhibited a significant increase in the tail flick latency compared to the baseline measurement. The latency time progressively extended after the drug administration, indicating analgesic activity, which means it has shown that mice, after administering the morphine drug, the morphine has exhibited significant increase in the tail flick. As we can see here, for the before administering the drug, it was the mean value was 3 seconds, but after administering for 15 minutes, the mean value was 5.3. Then for 30 minutes, it has increased to 7.4, and for 60 minutes, the mean value has increased to 9.17 compared to the baseline measurement, which means that the latency time progressively extended. The latency time has been extended after drug administration, indicating analgesic activity of the morphine. Now, dose response relationship. We have observed a dose dependent effect, higher morphine doses correlated with prolonged tail flick latency, which means this chart shows that this blue graph has shown that 3 seconds mean value, which means graph shows without morphine and the red line shows with morphine. So it has shown. We can understand with the help of this bar graph that by increasing in the time and increasing in the dose of the by increasing the dose of the morphine, we can increase the tail flick latency. The optimal dose for the pain relief without adverse effects warrants further investigation. And by this, we can calculate the optimal dose for the pain relief through the morphine through this experiment. So now we have learned a lot and we have learned about the tail flick method. We have understood the therapeutic activity of the morphine drug by its analgesic activity by performing the tail flick method through the analgesiometer. I hope that you have learned a lot about this in this experiment. So now we have completed our experiment. Thank you guys for watching this video and for more such learning content, subscribe to our YouTube channel RJPT Informatics. You can also follow us on our LinkedIn profile, on Instagram as well as in Facebook. We update and upload our such learning content over these platforms. Thank you.